Hi everyone, I'm Caressa, and over the course of this school year, I've been working with Dr. Shapira at the Structural Genomics Consortium on the data compilation of untargeted human proteins for AI-driven drug discovery. In this presentation, I'll provide the rationale behind this project, followed by the methodology, results, and some concluding statements. Starting off with some background, the completion of the Human Genome Project in 2003 was a groundbreaking milestone in the field of genomics and created high expectations for a surge in drug discovery. However, despite this breakthrough, the translation of genomic knowledge into effective therapeutics has been slow, with only 5 to 10% of proteins being successfully targeted by approved drugs. Moreover, the majority of the proteome remains understudied, leaving many proteins with poorly understood functions. This is in part due to a lack of change in research patterns since the Human Genome Project. Research and drug discovery have primarily been limited to 5,000 well-studied proteins, making up 95% of research publications, in which 75% of these still focus on genes characterized prior to the Human Genome Project. The consequence of this narrow study focus is that many possible disease-modifying proteins remain uncharacterized, including their roles in disease pathology and etiology. This limitation confines drug development to a narrow range of molecular pathways and mechanisms, slowing the development of more effective and precise therapeutics. This lack of innovation is reflected in the pharmaceutical industry's drug approval rate being at its lowest point in the past decade. Only 11% of compounds from first-in-human studies become registered, with the major cause of failure at all stages of drug development being insufficient efficacy. These failure rates have not changed since the 1990s and highlights a major concern with the lack of innovation. Additionally, stage 3 trials have a failure rate of 43%, with 66% of these failures attributed to insufficient efficacy. These failure rates in late-stage drug development are what contribute to the high cost of bringing a drug to market, both in monetary and resource costs. And it also shows that established pharmaceutical targets may no longer provide therapeutics with efficacies that justify their high developmental costs. In turn, it underscores the need for identifying novel pharmaceutical targets to overcome the static nature of bringing therapeutics to the market. In a more positive light, there have been major advancements in drug discovery tools, such as large-scale chemical libraries, high-throughput screening, and functional proteomics. Most notably, the development of chemical probes has created an efficient method to determine a protein's mechanistic and phenotypic role in various study models, and it can also provide information on the clinical translatability of a target. High-throughput screening has greatly contributed to the discovery of small molecules and chemical probes, and has also benefited from the development of high-quality probes for lead target discovery. Newer approaches, such as DNA-encoded library screening and affinity selection mass spectrometry, can now identify probes for more complex drug actions outside of traditional enzymatic functions. And it can also handle larger compound collections than classical high-throughput screening. In addition, recent combinations of these screening tools with AI softwares have improved hit identification, predictive accuracy, and reduced false positives to accelerate the discovery of novel therapeutics. Yet despite these advancements, research bias for well-studied pharmacological targets remains. This lack of proteome exploration and established efficiency of new technologies led to the development of Target 2035. This project collaborative between academia, industry, and funders aims to develop cell-active, well-characterized probes and chemogenomic libraries for the entire human proteome. As an open science initiative, these tools will be publicly available to encourage exploration of the unexplored proteome and result in a greater understanding of protein biology and their impact on human health and disease. This, in turn, can catalyze the discovery of novel therapeutics. Drug screening technologies and AI combination systems have previously been developed, however, are not publicly available. This also includes access to large-scale and comprehensive data needed for training AI. Thus, this project aims to contribute to Target 2035 by creating a dataset of all human proteins with no known exogenous ligand for training future AI or machine learning drug discovery platforms, including creating standardized DEL and ASMS screening protocols. This in turn can accelerate the development of ligands and probes for these untargeted proteins to advance the discovery of novel therapeutics and further our understanding of human biology. This is an overview of the methodology I've developed to extract and compile this dataset. It's split into three sections with an end goal of having proteins that are in the protein database, or PDB, with no known exogenous ligand. This emphasis on proteins in the PDB is because these proteins have been efficiently cloned, expressed, and purified, which is necessary for ligand discovery analysis. The first step, highlighted in green, is to define all ligands in the PDB as either endogenous or exogenous, and this was done using the database Reactome. The second step in red is to use the ChemBioPort database to identify human proteins in the PDB. And the last step in orange is to identify what ligands are bound to a given protein before using the list of endogenous ligands from the first step to identify proteins with no known exogenous ligand. As I mentioned, the first step is to identify endogenous and exogenous ligands in the PDB, and this was done using a second database called Reactome. Reactome provides a comprehensive map of the different normal physiological functions in humans, and it also includes the ligands or molecules involved in these processes. So using this database, all the ligands in Reactome were downloaded, which are provided as their Kebi IDs, and this is used to identify endogenous ligands. Then, all ligands in the PDB were downloaded from Ligand Expo, which is the PDB's database for their ligands. And this dataset also includes all PDB entries bound or associated to each given ligand. And these ligands are provided as their CCD, or Chemical Compound Dictionary, which is the PDB's identification system for their ligands. Because the reactome ligands are listed as their Kebi IDs and the PDB ligands as their CCD IDs, I converted both datasets to include each ligand's INCHI ID, which is the most accessible identification system used by both databases. The PDB already provides a file with the conversions to INCHI, and the Kebi database also has a file that converts Kebi to INCHI. So using these INCHI IDs, a Python script was created to identify overlap between the Reactome dataset and the PDB dataset based on INCHI. Any PDB ligand that overlapped with the Reactome dataset was deemed endogenous, and the rest was cataloged in a separate dataset for exogenous ligands. So at the end of this step, there is a dataset of all endogenous ligands in the PDB and their associated PDB entries. The second step is to identify human proteins in the PDB, which was done using ChemBioPort, an online database that can provide information on all human proteins, including all of their associated PDB entries. And in this data set, it includes the ChemBioPort identifier, the protein's UniProt, and all PDB entries associated with this protein. So using this database, I was able to obtain a list of all PDB entries for a given human protein. The last step is to identify what protein is bound to what ligand and keep the proteins bound to an endogenous ligand. This was done by extracting the API of each human PDB entry to split each entry into its chains and then identify what ligand is bound to what chain. So this is a snapshot of a PDB entries API showing the information for chain A, which is bound to ligand LZ9. The API also includes information on gene name, chain type, and amino acid sequence. The reason why each entry is looked at chain by chain is because a given PDB entry is a crystallized structure. However, it is not necessarily one singular protein. An entry may be a protein bound to a non-protein component like DNA, or be an interaction of multiple proteins. Because of this, there may be situations where an exogenous ligand is bound to the non-protein component or only one of the proteins in the complex. I've shown an example here of PDB entry 5HQ0, which is a ternary complex of CDK1, cyclin B, and CKS2. By extracting the different chains of this entry, it can be identified that only chain A, which is CDK1, is bound to an inhibitor. In these cases, looking at what a PDB entry is bound to as a whole is not specific enough in identifying what a given protein is bound to. This step was done using KNIME, and I'll be going over a simplified version of the workflow in two parts. First, the list of all human PDB entries obtained using ChemBioPort was loaded into the system. Then, the PDB IDs are appended to the end of the base API link. So you'll notice here that 5HQ0 is appended or attached to the end of the base link leading to the API. This allows the API of each PDB entry to be extracted and then placed in a table. Then two filter nodes separates the information of the API relevant to the chains and the ligands. For the chains, the table contains the PDB entry, the chain ID, what type of chain it is, and the gene name. For the ligands, the table contains the ligand's CCD, what chain the ligand is bound to, and what PDB entry it is from. Following this, only the protein or polypeptide chains are kept, and water is removed from being identified as a ligand because of how ubiquitous it is. However, this does not exclude water-bound proteins. Then the information from these two tables are merged. After this stage, the dataset of endogenous ligands, which was created in step 1 using Reactome, is loaded into the system and used to separate chains bound to endogenous ligands based on overlap. Chains that did not overlap were placed in a second table containing both exogenous bound and unbound proteins. This table is then separated into chains bound to an exogenous ligand and chains bound to nothing. The endogenous bound proteins and unbound proteins were then double-checked to make sure that they don't occur in the exogenous bound protein list, and these proteins were checked based on gene name. Then, the endogenous bound and unbound proteins were merged into a final table. This final table contains all human proteins in the PDB that don't have an exogenous ligand. This is a section of the final dataset, which contains the endogenous ligand, if any, the PDB entry, UniProt ID, gene name, chain type and name, amino acid sequence, start and end amino acid, and finally the chain ID. To put some numerical perspectives into the results, around 45 million endogenous ligands were found in Reactome, and out of the 42,000 ligands in the PDB, only 281 endogenous ligands were identified. This is less than the true amount of endogenous ligands in the PDB, which will be discussed in the limitations. In regards to the proteins, 11,000 human proteins were identified from ChemBioPort, with 8,000 having PDB entries. The final dataset contains 7,141 unique proteins that have no known exogenous ligand and are of interest for further research. There are two main limitations to this methodology. The first is incomplete identification of endogenous ligands in the PDB, as previously mentioned. This is because Reactome documents ligands in the state that is relevant to physiological processes, which are often different charged states. However, the PDB documents only one version of a given molecule in its most common form. For example, Reactome documents ATP with its charge of -4, which has a Kebi ID of 30616, while the PDB only lists it as ATP without the charge, which has a Kebi ID of 15422. This difference is also true for the given INCHI IDs. Thus, there are endogenous ligands in the PDB that couldn't be recognized by the script. The second limitation is that remnants of chemicals used during experiments, such as solvents and buffers, are sometimes still bound to the crystallized structure. Even though these are exogenous, they do not serve any modulatory or pharmaceutical function, thus should not be the eliminating factor for a protein. In this example, 3WB8, it is only bound to a ligand called 1,2-ethane-diol, which is used as a solvent and cryoprotectant when freezing biological samples. Because of this chemical, 3WB8 was incorrectly removed from the final list. The next steps are to, of course, refine this data set by more comprehensively identifying endogenous ligands in the PDB. A possible method is to use the IUPAC naming system rather than INCHI when looking for overlap, because IUPAC normally does not differentiate charged states for biomolecules like nucleotide triphosphates, which are the main group of endogenous ligands excluded. So for instance, ATP and ATP4- or 3- have the same IUPAC name. Secondly, the presence of solvents and buffers need to be addressed and removed from being defined as an exogenous ligand. And this may be done by looking at manufacturing websites, which provide lists of their products and their different IDs, such as Kebi, INCHI, and IUPAC. There are also literature reports of solvents commonly seen in protein structures. The long-term goal of this project is to create new AI or machine learning platforms trained on these untargeted proteins for drug discovery. As mentioned in the intro, DEL and ASMS are screening technologies that can handle large compound collections and identify more complex drug actions like proteolysis targeting chimeras or PROTACs. So to integrate these screening tools into this platform, standardized DEL and ASMS protocols need to be developed. Overall, this methodology establishes a dataset that identifies proteins with no known exogenous ligands, creating a foundational resource for AI-driven computational modeling in drug discovery. By cataloging these understudied proteins, this dataset enables the development of models that can assess potential ligand interactions, aiding in the identification of novel drug targets that have historically been overlooked. Given the high failure rates of conventional drug discovery, particularly due to insufficient efficacy in late-stage clinical trials, this approach presents an opportunity to diversify and optimize therapeutic development by expanding research beyond well-characterized proteins. Furthermore, these findings contribute to the objectives of Target 2035, an initiative dedicated to developing publicly available chemogenomic libraries for mapping the entire human proteome. By highlighting these untargeted proteins in need of chemical probes, this work supports future efforts for more comprehensively understanding protein function in both health and disease. Chemical probes serve as essential tools for validating drug targets, elucidating biological pathways, and uncovering mechanisms of action, ultimately accelerating the transition from basic research to translational medicine. The intent on integrating AI-based computational modeling with this dataset also enhances the efficiency and accuracy of target identification by reducing false positives and optimizing compound screening strategies. This collaboration between AI and experimental validation streamlines the discovery of small molecules with therapeutic potential, addressing the persistent challenge of limited innovation in drug development. Moreover, by making these resources publicly accessible, the initiative promotes collaborative research, ensuring that advancements in proteome exploration benefit the broader scientific and pharmaceutical communities. In summary, this methodology not only advances AI-driven drug discovery, but also aligns with global efforts to increase access to chemical biology tools. By systematically identifying and characterizing previously untargeted proteins, this work has the potential to improve drug discovery pipelines, identify novel therapeutic opportunities, and accelerate the development of more precise and effective treatments. I'd also like to acknowledge Dr. Shapira for providing this research opportunity and my two graduate supervisors, Angela and Luca, for guiding me in this project.